Thank you all for standing by, and welcome to our webinar entitled Monitoring Microcystin Production Rates in Western Lake Erie. This is a webinar series called Freshwater Science that highlights Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Benicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Justin Chaffin from Ohio State University Stone Laboratory. Dr. Chaffin is a senior researcher and research coordinator at Stone Lab within the College of Food, Agricultural, and Environmental Sciences here at Ohio State. His research interest is Lake Erie phytoplankton ecology, with particular interest in cyanobacterial blooms. His recent and ongoing research projects include linking experiments and models to predict cyanobacterial bloom toxicity, determining drivers of benthic algal blooms, and the effectiveness of data buoys at measuring cyanobacterial bloom biomass, and rapid microcystin tests. We're delighted to have Dr. Chaffin here today to discuss his research. Before we do, just a few things about the webinars themselves. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20-ish or so, I'll conduct a question and answer session. If you would like to ask a question during the presentation, please feel free to pull up that chat feature located at the bottom of your screen at any time during Dr. Chaffin's talk, and I will collect and pose your questions out to him at the end of his presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Justin Chaffin from Ohio State Stone Lab, presenting monitoring microcystin production rates in Western Lake Erie.
Oh yeah, thanks, Jill, for that long introduction, and thanks everyone for dealing with the rescheduling. So this afternoon, I'm going to talk about a project we've been working on for the past couple of years with the University of Toledo. When most researchers go out to a body of water to study it, to study the blooms, to study water quality, we simply grab water samples and measure what's in them, looking at snapshot concentrations of algae or toxins or nutrients. But with this project, we wanted to parallel that traditional lake monitoring style with incorporating experimental work to measure how fast Microcystis is producing the cyanotoxin microcystin. So monitoring the concentration of the toxin, but also monitoring how fast it's being produced. So first, I always like to start off with some general overview slides about algae. So most algae are not bad for lakes. Algae are tiny plant-like organisms, and half the Earth's oxygen is produced by algae. So every other breath you take, you should thank algae. Algae are the base of the food web. We have algae come in all different sizes, shapes, and colors. The beneficial types of algae are consumed by zooplankton, and then the zooplankton are consumed by small fish, and small fish are consumed by large fish. However, too much of the wrong type of algae is harmful. You often hear the term harmful algal bloom. When we're in freshwater, the main drivers of blooms is excess phosphorus and nitrogen. And it doesn't really, the algae don't care if it's coming from fertilizers or manure or lawn care or untreated sewage. If it's put on land to grow crops or to grow grass or plants, it will grow algae once it gets into the waters. Some problems associated with the blooms, they will produce these secondary compounds that have toxic effects. So there can be human health issues. The toxins can get into drinking water, and we're learning a lot about how these toxins can get into the air and cause inflammation in lungs. A lot of pets or wildlife have died associated with toxic blooms. Um, the cyanobacteria are not really consumed by zooplankton, and when they are, they don't reach the same biomass, and the good type of algae dies off. So there's negative ecological effects, and I think we got to do a better job of documenting the negative economic impacts that these blooms can have. For example, property values can decrease. Uh, the and It costs a lot to treat drinking water to remove the toxins, and it can be a large tourism hit associated with blooms. So the blooms in Lake Erie are a type of algae called cyanobacteria, also called harmful algal blooms. The genera is Microcystis. Microcystis produces the cyanotoxin microcystin. The blooms always occur in the western basin of the lake, so near the city of Toledo, near the outflow of the Maumee River, the western basin. These blooms are visible from space. They can be quantified by space, by satellites in space, and they occur during the summertime when the water's at its warmest. In Lake Erie, we can forecast the biomass of the blooms, but not yet the toxins. On a seasonal scale, we can forecast how green the water is going to be, how much biomass is going to be in the western basin, based on the phosphorus load from the Maumee River during the springtime. When there's more phosphorus getting into the lake, the bloom is larger. And the amount of phosphorus that gets into the lake via the Maumee River is highly correlated with the amount of rain we get during the spring. In this graph, although it's somewhat outdated, it still holds. 2011, 2015, and 13, these were very wet springs. We had a lot of phosphorus load, large bloom. 2012 was a drought. Drought, very little phosphorus load, smaller bloom. So we can use this information to forecast biomass, but not toxins. And then on a more fine scale approach, we can take remote sensing data from satellites that see the biomass, for example, here, and then we can plug that biomass into lake circulation models to forecast where that bloom biomass is going to be several days in advance. These models are updated basically daily, and then NOAA sends out these bulletins twice a week saying, here's where the bloom is, here's where it's going to be. Okay, but these bulletins do not work for microcystins yet. So an obvious question, is there a relationship between bloom biomass and microcystin concentration? So can you look at a water that is green and assume it's super toxic or not? So this is data that that my lab's been collecting for several years in the Western Basin. We have cyanobacteria biomass on the x-axis here and total microcystin concentrations on the y. And when we zoom into where most of the data lie, we see that there's really no correlation between the two. So if we're trying to forecast bloom toxicity or microcystin concentrations, we cannot simply rely on biomass. So for Lake Erie, we can forecast the biomass, but not yet microcystins. We know that phosphorus is the main driver for freshwater blooms, so more phosphorus leads to more algae. However, for microcystins, there's a lot of other factors that drive toxicity. Nitrogen is a really main factor. Light levels, changes in temperature all affect microcystin production, and there's even genetic differences among strains within the lake. Despite all that complexity with what triggers microcystin production, we have been making some progress at developing microcystin models. And there's a couple types of models that have been developed or are still works in progress. Some models simply ask, what are the chances of microcystin exceeding 6 micrograms per liter, which is a level for beach advisory. So well, what's the chances of microcystin being above 6 and low to high scale? These models rely on remote sensing data for biomass. They rely on researchers grabbing water samples and measuring the chlorophyll concentration and microcystin concentrations in that water sample. And then they apply these data to water circulation models, then to predict the probability of microcystins in seeding 6. For this example, on August 7th, 2019, high probability of seeding 6 occurred in Maumee Bay and along the Ohio shoreline, and decreased further out into the lake. So, what's the probability of exceeding 6 or not? Another model asked the question, would microcystins increase or decrease or stay the same week to week? These models rely on microcystin grab sample data. These models rely on microcystin production rates and also the same water current models for future to determine where those toxins are going to be pushed to. And these models show that when you include microcystin production in those models, we can forecast the extremely high concentrations better than models that did not include microcystin production in those models. And so, we're factoring biology into these models. And with microcystin production rates, these are data from 2018 and 19 that we have a more limited data set on. But we see that production decrease throughout from July through into October. So we have the highest production occurring in July and lower production occurring in September and October. So this project that we've been working on is monitoring microcystin production rates. So question, why do we want to monitor production rates? Well, we know that models that included microcystin production rates were more accurate than models without microcystin production rates. We know that including microcystin production rates into those models is needed to forecast those extremely high concentrations that might occur in Maumee Bay or along the shorelines. There are a lot of researchers that are using grab samples to quantify microcystin concentrations in the lake. However, it's difficult to predict the future concentrations based on that grab sample alone. So if we know if the cyanobacteria in those water samples are producing microcystin fast, then we can predict that microcystin concentrations will in fact go up. And also, if we're understanding what triggers microcystin production, what triggers cyanobacteria growth rates, and what drives them, this will be critical to understanding how the lake might respond to future conditions or any management actions that we take. So we've been working on this last year, 2024, and we'll continue it this upcoming summer. Us and the University of Toledo are sampling these sites in the Western Basin. The sites further east a little bit less frequently, but we're sampling these sites about at least once every 10 days during the bloom season, collecting all the regular grab sample data. And then we're at these two sites that I have circled, one in Maumee Bay and one in the center of the basin. We're doing incubation experiments to monitor microcystin production rates. And the next slide, I'll show you how we're doing that. Site NB18 and WB83 are the focal sites, which we're doing, again, I said the weekly microcystin production rates. We're also getting data on microcystin congeners and another type of toxins that we're learning about, the aminopeptins. And then during our weekly or every 10-day cruises, we're grabbing water from one other site that has high biomass, including that. So we're doing at least 4 incubations per every 10 days. So if we just look at the snapshot concentrations that we get into the routine water quality monitoring, we see that microcystins on the top are much higher in Maumee Bay than they are in the offshore. That's expected. Um, and cyanobacteria concentrations, again, much higher in Maumee Bay than the offshore. Again, that's expected. The bloom last year was very long. We had over 3 months of detectable microcystins at all sites. We had detection starting in late June, persisting into late October. This high peak in Maumee Bay on September 5 was collected during the scum sample, although our samples do account for scums and not being scum. So it was a depth integrated sample. So there was a lot of bloom biomass being pushed into Maumee Bay at this time. For our microcystin production rates, we are bringing water from the lake back to the lab and incubating it. We will put the lake water in these bottles, and for 3 of them, we'll leave them alone. So it's an ambient nutrient control. And for another 3, we'll spike in phosphorus and nitrogen. They're both important. Then we'll cap the bottles and deploy them off our docks and allow them to incubate for 3 days. And then we'll monitor the microcystin concentration in these bottles. And then when we make plots of concentration versus time, the slope of this line is the microcystin production rate. For example, you see in this graph, in the Maumee Bay site in late August, the production rate was 0.4 per day. And when you do the math for this type of slope here, production rate of 0.7 is equivalent to 1 doubling per day. And when we do this, we also track the cyanobacteria biomass and the other types of algae. So we can see how fast the algae are growing, but also how fast microcystins are being produced. So what did we find last year I'm really going to focus in on just the Maumee Bay site here. We see that, so here I have production rate on the y-axis. So numbers that are positive are concentrations are going up, they're increasing, and the negative numbers indicate concentrations went down. The white circles indicate the ambient nutrient control, and the black circles are the elevated nutrients. So we see that the elevated nutrients stimulates microcystin production in most experiments, but not all experiments, but most experiments, when we give them more nutrients, they increase toxin concentration. Last year, we found that the highest production rates occurred during the early bloom phases, which aligned with our 2018 and 2019 data. We found lowest production late bloom, again, which matched our previous data. And mid bloom, there was variable production. And what we are really diving into now is trying to understand why some of these dates had higher production, whereas some of them had lower production. Okay. So we're currently investigating a whole suite of environmental parameters, lake data and weather data to look at these. So I want to point out what we observed low production rates in late June, and we observed these low production rates across all our sites in late June. All the different icons up here are the different sites. I don't have them listed out, but you can just see the general pattern of higher production rates mid-June, dropping to low production rates mid-June, early July, and then variable and then decreasing again. So when we look at that, and one factor we want to look at is light intensity. So if we look at the amount of sunlight that was hitting the Western Basin, we see that there was a lot of cloudy days late June and early July. And this aligns pretty well with the lower microcystin production rates that we measured in our experiments. And we know that microcystis likes high light levels. It needs high light levels to grow and high light levels to produce microcystin. So when these cloudier days, it could have been hampering microcystin production. Although, again, we're looking into a lot of other factors too. So the question that we get, could get is what was the actual microcystin production rate, right? So we're measuring ambient nutrients and elevated nutrient concentrations. We don't know what the actual one is. And if we want to take a rate and put it in the model, we ideally want one rate. So our rates were measured in bottles without and with elevated nitrogen and phosphorus. These rates likely represent the lowest and highest potential rate that we would measure it in the lake. So the actual rate is probably somewhere in between the high and the low. For our modeling study, we simply just took the midpoint between the two. So if we have this graph here where we look at the elevated nutrients and ambient nutrients, and we just simply take the midpoint between the two, this is the production rates that we're looking at here. Okay, so now if we take those production rates and overlay the microcystin concentration, we can start to look for trends. And here I have the Maumee Bay site, and then the rates that we measured in the center of the Western Basin. And note that the microcystin concentration scales are different between the two sites. Okay, the production rate scales are the same, but the concentration scales are different. So there's some good agreement between the trends. For example, here I have highlighted when concentrations went up between two sample dates and when microcystin production rate increased. So if you look at here, concentrations, the stars increased, production rate was increasing. Production rate concentrations increased from this site to this, or this date to this date, and so did production rates. And we see these trends agreeing here and over here. And we also see when the trends were going down. Concentrations decreased, production rates also decreased. Okay, so there's some good agreement. However, the concentrations that we measure in the lake is also a function of water currents. Even though they might be producing a lot in one area, if they're being pushed, if we're getting a strong east wind, for example, concentrations can increase in Maumee Bay simply due to more biomass and more toxins being pushed that way. So again, the models will factor in the water currents there. So to work to continue, we are currently quantifying the phytoplankton community in our samples to see what cyanobacteria were out there. It was predominantly Microcystis, most of the bloom. We did see some Dolichospermum later in the season, but Dolichospermum doesn't produce Microcystis. Repeating the study again, and then compare microcystin production, also cyanobacteria growth rates to the nutrient concentrations, the water temperature, changes in temperature, pH, and sunlight, and other weather parameters. So the goals and outcomes of this. So we know that it's not feasible to measure microcystin production rates to form an operational forecast. When we're monitoring these production rates, we have to collect the water, incubate it for 3 days, and analyze those samples. That can take some time. By the time that process has completed, the forecast has come and gone, right? If we collect samples on Monday and we want to forecast concentrations on Tuesday or Wednesday, well, we're not getting our production rate data until after Thursday or Friday, right? So we're behind on that. So ideally, what we get from this work is that we learn a microcystin production rates that can be used to inform future models. For example, maybe we'll figure a predetermined production rate for each month, which might be higher early summer and lower during the fall. And then if we factoring in the environmental parameters, if we know that it's going to be cloudy, and if that pattern holds that when it was cloudy or we had lower production rates, then we could simply subtract or divide the production rates that we would recommend for the month of July, divided by half, or subtract X from them. And then those rates could then be used to put into the model. So yeah, that's the goal for us to monitor microcystin production. It's not to inform models, but it's to collect the data now to help build into future models. Besides that, I'd like to thank the US EPA, the Great Lakes Restoration Initiative, and the Ohio Lake Erie Commission for managing the funds, the Ocean and Human Health Program from NSF and NIH, the staff and students at OSU Stone Lab and University of Toledo Lake Erie Center, and the Judy Westrick Lab at Wayne State for doing the mass spec data for congeners. And with that, I'll be happy to take questions.
All right, we got quite a few questions, so let me just start rolling here. Okay, the first question we had was, when you refer to microcystin, is it measured as total microcystin or solely microcystin LR?
So the data that I presented was through the ELISA method, which measures microcystin LR equivalents. So it measures total microcystin, but depending on the microcystin content profiles, it might be skewed a little bit, but we're measuring total microcystin, assuming it's LR equivalents.
Okay.
I could go into a lot longer detail, but let's just say it's total microcystins. Yes.
All right.
Not
And
just one congener.
I've got a couple more clarifying questions too. Was chlorophyll A used to track cyanobacteria biomass?
Yeah, so the chlorophyll A we use is from a fluoroprobe, which measures green algae, cyanobacteria, diatoms, and cryptophytes. We take those data and then apply a correction factor to the water extracted chlorophyll that we use. So if total chlorophyll's 15, and if we know half that chlorophyll is from cyanobacteria, then we assume that half of 15 is 7.5, and that's the cyanobacteria biomass.
Do dissolved oxygen levels affect the production?
Not really.
Okay.
Let's just keep it. There's some
All right.
caveats, but not really.
All right. Well, another question I got was, I understand microcystins are released when cells die. Do you know of any work or data correlating or predicting algal die-offs with increased toxins?
Yes. So the other half of this project is looking at what triggers cells being lysed. I didn't talk about any of that, but all our samples, we're also measuring extracellular microcystins to see when do extracellular microcystins, when are they present, and what are the environmental factors associated with those. We know that it's likely due to viruses attacking the bloom, but still yet trying to figure out what triggers a virus infection.
All right. Another question that we had gotten was, is there a measurement system that can measure microcystin without pulling water samples? Just wondering about things like bloom measurements done from space, some kind of measurement system that that could be used outside of pulling water samples.
Unfortunately, no, because there is no direct correlation between algae biomass or how green the water is or whatever parameter you want to look at and microcystins. So majority of the microcystins are inside a cell. Satellites can't see through the cell to see microcystin. So no, to monitor microcystins, you have to have a water sample.
All right. Do MC concentrations correlate with any other environmental, like river flow, meteorological, or community changes, either microbes or phytoplankton or zooplankton?
No, there's a lot of there's no direct correlation between most of those things, but they do play a role. For example, we know that nitrogen has a a strong limiting role. Early summer, there's a lot of nitrate in the water. There's lower biomass in the water. There's more sunlight. The water's clear. Those things trigger uh, microcystis cells to produce more microcystins per cell. Later in the summer into the fall, when there's very little nitrate, uh, the microcystis needs nitrogen to produce microcystins because it's a amino acid that has a lot of nitrogen in it. Um, when there's very little nitrogen, toxins are lower, but also light is lower as well. So no, there's not just a simple correlation between X and microcystins.
Okay. Um, we just to clarify, because I think you had talked about this earlier in your presentation, and we got a question just clarifying, is there a correlation between algal density and increased level of microcystin?
No,
Okay.
there's no correlation. You can have green water, super green water that has low toxins, and you can have water that is just has a little bit of green but have high concentrations of microcystins.
All right. Another question that we got was, given that strain genetics can play a role in determining microcystin production, do you think that near real-time information on microcystin toxin genes and or their expression might be a useful addition to the model?
Uh, yes, but we can at least with our current methods we can go from gram sample to microcystin concentrations in 8 hours, running at the fastest we can. Um, so yeah, the near real time genetic stuff is going to be powerful. Um, it's going to be expensive, but it would have to be used broadly throughout the lake. So I think that's the challenge is having enough spatial data to inform the model.
Another question that we had gotten was, one of our participants was interested in why 3 days was chosen as the incubation period. Did you look at production rates calculated based on shorter and or longer incubation times?
Yeah, that is a good question. So on shorter scales, there are sometimes, well, let me just ask the longer. The longer you keep a community trapped in a bottle, things can start deviating too much. Other things can become limiting, such as carbon. Um, Zooplankton can build up, and sometimes if there's lots of zooplankton, they'll start eating things, and then they start excreting waste. So keeping them shorter is better, but too short, we might not see a response yet. So between 2 to 3 days is ideal.
Another question was, we've got a few more if you're okay with
Yeah,
staying
I'm
on
fine.
for just a little bit longer. At our lake, we're noticing higher concentrations of nitrogen, phosphorus, and chlorophyll at about 3 to 4 meter level. Have you noticed different concentrations in your studies as well?
I'm assuming that question is about going down
I think so.
through the profile. So at 3 meters and 4 meters down, you're seeing a spike in algae and nutrients. Without knowing what lake or seeing any other things about that lake, I'm going to assume that there's what's called uh, there couple things. It could be it could be a bloom of planktothrix hanging out at the thermocline, but you could also have nutrients coming up from the bottom, the planktothrix hanging out right at the thermocline and using it. But again, without really knowing for sure, um, those are just guesses.
Okay. A few more questions. Um, Do we know why cyanobacteria produce toxins, or is it still a mystery?
There's some theories for microcystins. The leading theory is that it helps the cells against oxidative stress. So when the uh, when there's a lot of sunlight, they're photosynthesizing a lot, but they're producing some free radicals, and having microcystins um, helps the cell get through that. And it's also supported by the community shifts that we see in the lake, where the conditions that select for toxic strands also encourage the formation of other free radicals. And then later in the summer, when there's not the high sunlight and lower free radicals, we have more non-toxic strains. So at least that's the current theory, is that it helps the cells get through oxidative stress.
Our last question, we've actually had several people ask about different lakes. If you know, like there was a question about if you know of anyone who is studying algal blooms at, like, say, Buckeye Lake.
Um, not not personally, but I know that Ohio EPA monitors things, or Division of Wildlife monitors those lakes. Um, I'm not sure if Buckeye Lake is, if I can't remember if it's large enough for remote sensing to see it, but
Mm.
um, Lake Erie is not the only lake that has blooms in Ohio. Any lake that has a lot of nutrients getting in it, phosphorus and nitrogen, and is warm enough during the summer, will have blooms.
All right. Well, I think those were the majority. I think I got all of them. We had quite a few questions, so thank you for staying on, and thank you everyone for your questions. And so let me just um, end the session with a big thank you again to Dr. Chaffin for his willingness to talk to us today about his algal research. It was really an excellent discussion, so thank you again for being here and talking with us. I did also want to remind everyone that we have our survey URL for this webinar in the chat feature, so if you could take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue on next month. We'll email you when that webinar registration is available. We hope this was beneficial and hope you'll join us again in an upcoming webinar. Thank you, everyone. Thank you, Dr. Chaffin. This was great. We really appreciate it. Everyone have a great afternoon. Thanks, Justin.
Thanks, everyone.